So we're cruising along, at least on the bathymetry, we're cruising along the northeast side of the ridge. And this, it's still got plenty of corals on it, although I can't say there's a huge abundance here. They're kind of in little groupings or patches. And they're kind of on these little high points sometimes too, at the big boulders or blocks or things like that, which we commonly see. Is that a sea star, Chris? Right here, next to the coral.
I bet it's a brittle star.
Top
It's
of
a
the
brittle
screen.
star? Oh.
Sorry. I bet it's one of those Okeacanthids.
Yeah, he probably just jumped off. Come on in.
Okay.
Oh,
he's still on it.
A coral.
Yeah.
Oh, in fact, it's still on.
Well,
How's it interacting with that? It's got a squat
Come on in.
monster.
Well, it's got some short legs
Oh, that's interesting.
on that one side of it.
Yeah, 3 of the arms have suffered sublethal predation.
But you think the crab is defending the coral, or if he's even doing
at least keeping it out of its way.
Yeah, that's what I was wondering, whether it's more of a competition interaction.
Yeah, let's keep an eye on that.
Or is it defending itself? Maybe the
Yeah, that's
guy is
possible. But
after the.
look, it's okay. Well, thanks, Brittle Star.
Now that's
I
what
think
you.
Jim Shank was noting earlier that we don't usually see this many Okeacanthids on Kartsvik Gorja as such as we've seen today. So that's an interesting observation in and of itself.
I was going to say, you just saw an Ophiacanthid ROV interaction. They commonly sort of jump off, but we don't know how often they do that in nature without any lights on them.
Yeah. Oh, I know.
It's impossible.
We started seeing it almost directly, I think, during Santiago's leg.
Do you think it's the lights, Chris, or the vibration?
It's possible.
That's all they're doing. They're climbing to the top. They jump off and they climb the top again.
But it would also make sense if they kind of had a sort of back and
coral
forth
on the end.
You could do a snap from.
Yeah, this is the one we collected again with the weird Paragorgia, Canthogorgia, Scleroxonia, whatever it is.
Tiny sponges down there.
Yeah, little tiny. These look like the kebab sponges, but there's only one piece of sponge on each stick.
I still personally like this for Paragorgia, so I'll be interested to see what the sclera is.
Oh, here's
There
another
you go.
Astrophyra.
All right, we're going to get
Right
a
in the
super
center.
close-up of this.
It's a brittle star, but we're looking at it right head on. Man, I love these
moving
cameras.
lights.
This is
We'll,
like 3
we'll see what it looks like.
ROV nav.
in situ.
Move complete.
So,
All right,
yeah.
come on in, Rolla. You center
Boom.
him up. Hold on. Gotta
Not a
move
sea
little
star, but a brittle star.
bit. Oh, come back out.
Okay, try that again.
Yeah, repositioning.
What's that? Yeah, maybe.
All right,
Let's see if.
come on in, Rolla.
It's these plates that are actually going to be useful for a diagnosis to species, if it's known. So this is Astrophyra. This is a brittle star. Notice the arms are really
Is
tiny.
that Max?
Maxa.
Yeah, and so this isn't the juvenile form of any other species. This is an adult, as far as we can tell. If you turned it over, it looks pretty much like a brittle star. And for some reason, they've developed this appearance that is convergent
Stay in
with
focus.
other sea stars that live flush on surfaces. We used to think that they lived on wood substrates because they're similar to wood sea stars that are kind of flush and flat and live on wood surfaces. But I guess this is the third incidence of seeing a living one in situ, and they live flush on these rocks. It's amazing. I've seen these before, but up until 3 days ago, I'd never seen them alive, and now we're seeing one just like it was just. These are like the size of a quarter and less. So I'm always bowled over when I see them because I usually see them in museum specimens, never alive, and because they're so freaking small. And now we can see the plate patterns. I'll have to work and see if I can figure out what species it is because that's just amazing. I could stare at this all day, but I'm sure everybody else would be bored.
Between the arms there, is that fleshy or is that platy and kind of hard like a typical ophiuroid?
I'm sorry, what was it? So everything that's in between is just plating. That entire thing is the disc.
Video's clear.
So he just has reduced arms, right? Is that what you're saying?
Yeah. So they just narrow and terminate right at the tip. See, things are looking up.
That's pretty weird.
Yeah, one of the reasons they have such a unique status is because they always end up in the textbooks as the example of weird ophiuroids.
Roger, Nav, go ahead.
Pretty pink.
Yeah.
Where
Sorry,
is it?
right at the base of this red coral, I think we got something different. So straight down, or is that? Oh, I think we could go directly upslope, frankly. We just didn't get that kind of detail in the multi-beam resolution that we were looking at. So, yeah, great, thank you.
I'm pretty much looking uphill at 100.
Scott, did you hear that? Is that pretty much your idea too? As we were just trying to keep going upslope toward the highest point of the seamount, and if the multi-beam was slightly off on, where we put our points, then we should just keep following the train and keep going up, correct? Or did you have another idea?
Nick, come on in, Roland.
Actually, I stepped out, but I just heard your question. And so, yeah, if we're on the top of the ridge, I say we just keep going in the direction that we started, right? So let's see, would that be, I don't remember if that's east or south. But yeah, just heading along the top, and local topography may change a little bit up or down, but I say we just stick with it. Maybe if the interest in the ridge isn't too wide, we sort of move from one side to the other.
Okay, thank you, Scott.
It looks like this, is this a bamboo coral? It's something that's been stripped of a lot of polyps, and I wonder, I don't see a sea
To the
star.
left, pilot, yeah, that one.
Anyway, I'm stepping aside for a couple of minutes to look after all the four-leggeds here and start my dinner. I'll be back a little bit later.
Okay, great, thank you, Scott.
What is this?
Keton math, I think.
Is that right in front of us?
Yeah.
That seems more maneuverable I gave than I thought.
It's actually right inside the camera lens.
Yeah. Come on in, Roland.
Well, it's a leggy bamboo. Internodal sparse of some kind, looks like. With a lot of long needles.
Guess the pets will have to wait.
Yeah.
Is this?
I can already see this one's got the prominent needles. So we've talked several times today about the sclerites in the shape of needles, which is common for this whole subfamily, the Keratoistidae, to which all the bamboo corals that we've been seeing on these dives belong. But this is one of the better examples that we've seen, and I think the only example we've seen today of these spines being so prominent. Now, the tentacles have been folded over. It's kind of like withdrawing your fingers into a fist. And we can see that there are some of these sclerites, they're very long, and they're at an oblique angle in the polyp body wall, and others are sticking through. And I wonder if Abby Lapointe has joined left. She might have something to say about this one.
Also point out that at least one of the arms on this ophiorid is regrowing, and I don't see a squat lobster, so squat lobsters may pinch off some of the arms on some of these, but there may be something else that's picking on these things too.
Sure. And squat lobsters are fairly unusual on the bamboo corals, but we know that these ophiuroids are very mobile, so they can move from colony to colony. So it may be that a squat lobster somewhere else was clipping off the arm, so we can't completely reject that. Oh, you see right under the arm there, you see there's something orange. I think there's a worm moving up. If you can go back down the colony here.
Oh, yeah, you zoom in right there.
Oh, yeah.
Yeah.
There's two of them. Yeah, so there's another one. So earlier I said I thought I saw one on the inside of a polyp. So I wonder if these are little flatworms or something. Can't tell. I guess it's on the outside of the bamboo coral, not the inside. And I guess you would like a clade for this. So give me a second and I'll give you a clade name. I'm not hearing from Abby or Les.
Let's maybe at lunch with Abby too, because it's about lunchtime right now.
Do you want to look to see if there's another worm elsewhere on it?
Hey guys, this is Tim. I'm just wondering if you're convinced that that worm is on the outside, or if you think it could be on the inside of the tissue.
And the one I saw earlier was certainly on the inside on a different colony, and I'm not sure if this is on the inside or the outside.
I sort of had the impression, looking at this one right now at the toward the bottom of the screen, that it's on the inside, but the one up top I wasn't sure about. Really interesting.
Yeah, it is. I don't know either, Tim.
Chris, did you notice, was this internodal or nodal branching?
I think it was internodal, Scott.
Yeah, come in, Roland.
Internodal, dang it.
Isn't this the bamboo
it's
coral that Les keeps on referring to as Wolverine?
Yeah, that's why I wondered if Abby was around. She's the one who came up with that particular name.
Okay, I think we're good. Pilot, I
Right,
think
come
we
on
can
out.
move on. So whatever is gluing those dead farreid sponges to the substrate, it's pretty good glue, because that's not a bad size sponge that's hanging off to the side of him, and yet that stalk isn't getting pulled over. The upper side of that colony is gone. I'm not really sure what the reason for that is, but this is one of these polyopogon sponges. In this case, it's not the yellow one, so I gather there's perhaps a couple different species
There's
that
another
are
sea
doing this.
cucumber, or maybe it was
Off my port pot.
the exact same one, but the one to the left there.
Ship is stopped, so Sirius should not be swinging right now.
Okay.
Right, come on in, video.
Yeah, I love these because you can see everything inside so clearly. You have some nice guts there. You have the tentacles coming off of the top, the mouth's facing down the bottom, and if we were to look at it from a slightly different angle, I'm sure we'd see this is the lower intestine, the upper intestine connected to and the stomach connected to the mouth, and we haven't seen any swimming sea cucumbers lately, which is unfortunate because they're so beautiful, and you can see the sort of dump the poop and take off into the water column behavior they're so well known for. That's lovely. Thank you.
This is the front face of one of these things. It looks like it's got a bunch of stuff on it. They're really beautiful, unique sponges. They don't look like anything else on the planet, if you ask me. A lot of deep sea sponges are like that.
So which one is this?
This is, again, I think it's Aspidoscopulia. It's in the Furiidae
that's
family.
Aspidoscopulia.
Well, it could be. This is what I was sent a paper by Dr. Konstantin Tabachnik of the Shirshov Institute in Moscow, and he worked on this genus. He published in 2011 a description of some of these, and based on the photographs, and it looks like it could be. And it's a tiny little, that's a baby one of these, I think. Just starting out.
It's interesting. There's that one next to it that looks the same size, but it's dead. I mean, it has some of those same side chambers, but
Yeah,
interesting.
right. And looking at another Chrysogorgia
Oh,
with
little
a
polychaete.
little polychaete.
All right.
All right, pilots, ready for another move, 100 degrees.
Yeah, I think so.
Copy that. I'll put another 30 meters in.
All right.
This is the bridge. Go ahead.
Ready for another ship move?
Yes, sir.
30 meters at 100 degrees.
Pilot,
0.2
there's
knots.
a stock sponge to the upper right.
Range 30
I think it
meters.
could be something
Bearing
different.
100 degrees. Speed 0.2.
Good copy.
Coming up.
That's this guy right here. So there's one type of Bolosomini that I haven't seen for quite a few years, and it has kind of a flatter ball on it. And that's the best we've been able to
come
go
on in,
on
Will.
this. We have a specimen of this already, and we still haven't. I mean, this could just. Oh, look
Ooh,
at that! A benthictinophore on it. Oh,
Benthictinophore. So benthictinophores are
We kill lasers.
Go for it.
You might be familiar with the swimming ctenophores, which are cnidarians. They swim using sort of little paddles called teen rows. They
now
capture
moving the
food
shade.
using their tentacles with little adhesive
Still
cells
drifting
called
forward.
with little adhesive cells. There's a subgroup of ctenophores which live on bottom, or which live on the bottom, or at least live on substrates, benthictinophores, and these have the same kind of general strategy. They extend these long feeding arms into the water. They're very unusual, and you'll often see them on sponges, and on some of them get quite big, relatively speaking. Some of the ones that we saw earlier in the shallows of Johnston Atoll
Try to follow
are
the
about
stringer.
4 to 6 inches tall. This one looks relatively small, but these tentacles can go on for quite a long while. We've, I think, followed these tentacles on others, and they've extended almost 3 feet in length, if not longer. And I see we're sort of following that same length. Some of the others that we've seen can actually have additional sort of projections coming off of the tentacles.
It's the end of it.
so I think
Yeah,
my
coming out.
sponge got ctenophore bombed there.
Coming back to the sponge.
You were pretty bored twice in a row. My sponge descriptions, you completely went on to your animals
Right,
there.
come on
so
in,
I
Roland.
thought I could finish my sponge thing here.
Come in
So
tight.
I think this is just a
I'm sorry, Chris.
I think this is just a morphological difference, and I think this is the Bolosomini, the ET sponge. And this is beautiful sponges, as you can see. And all of that sort of meshwork inside of it is part of the skeletal arrangement. And these are glued together or loose fused spicules. And yeah, I can see clearly by the texture that this is another one of these Bolosoma new genus. We don't have a name for it yet. Thank you very much, pilot. That's fine.
There's an even bigger
I
one.
humbly and dearly apologize to Chris on the air for having interrupted his
Twice.
twice
On
interrupting
sponges.
his extremely noble and informative narrative on these fascinating glass sponges.
You're not giving me a chance to talk about sponges here, but that's okay. Anyway,
I apologize.
no,
Before you
that's fine. It's fine.
I'll let you interrupt. I would let you interrupt me, but the ship does that already. So
Let's get this guy swimming.
oh, neat.
Go ahead in.
We kill lasers.
Yep.
A little hydrozoan, maybe a hydromedusa.
Yeah. Those little forearms.
He's close to you.
It's too transparent.
Yeah, there's no color to it.
Yeah, coming tight.
That'll make the midwater people happy. Lots of cool little jellies.
Yeah, let him go.
Something that we haven't named or said today is that we have named our cruise for this particular cruise, and it
It's
really
getting busy
applies
up here.
to all of the Okeanos cruise
Oh, there's another thing.
cruises as Laulima Okamoana, which is sort of a
Let's go. Let him
Hawaiian
go.
term for partnership in the seas. And like I said, Laulima literally means many hands, typically used in the context of many people coming together to make something
That
happen.
star-shaped thing
And
just
Laulima
past the big sponge on the
basically
right.
symbolizes
base.
cooperation,
Come in, rolling.
unity, and a shared vision. So all of us that are helping out and
Yeah.
coming together
Base.
to make this.
There was something white on that sponge there. There we are. That yellow
Unida.
sponge might have a. Is it a squat
Come on in.
monster?
Yeah, but it's anyway, beautiful view.
Okay.
Trying to read down on some of the points. I thought we were going to have a break, and then I look up and there's a whole bunch of animals everywhere.
We've reached a zone where it's getting very crowded.
That's good news.
Still drifting in.
Yep.
Yeah. When you're done. Thank you.
All right, rolling. Just let him go when you're good.
Okay, I think he's Munidopsis. And there's a lot of stuff on this
Different one on the
little
right.
high point here.
Right next to lasers.
Quite a bit of stuff up here,
On
huh?
the northern side, I think. Of this feature, I have no idea where we're just going to go up on a
Sponge
mountain
in
and then come
HC2
back down
you.
again.
Oh, never
but
mind. You're above it.
pretty steep right here. Wow, that's big. I wonder if we could take a look at
There
that.
is a huge sponge
Yeah.
to my left.
That's what I was looking at.
Yeah.
Wow. Looks dead though, huh?
Yeah, and there's another one of those yellow polyopogon, these weird ones.
It's a beautiful overview. If you
Jeez.
maybe tilt up a little
Yeah,
bit.
you're
Talk
right.
about a Dr. Seuss
Kill lasers.
like landscape. Wow.
Look at this. This is amazing.
That
If we could just hold the scene for a moment.
Wow.
Yeah, just do your little pirouette
It's
thing.
like the real land of Dr. Seuss and magical things.
This is Attack of the Sponges here.
You wanted sponges, Chris.
Yeah, I got them. Yeah, that's, well, okay, I got them. Wow.
So what is some of these I recognize, but for example, these big sort of,
Can you bring my uppers
the
in
ones
a
that
little
look
bit?
like
Yeah.
like amphidromes, the big half cups, are those anything you recognize?
Well, it could be furriids, and it also could be something that's called Conolasmatinae new genus. It's a little bit
There's level
controversial
coming in
right
more.
now.
Yeah, that's good.
Okay.
There's discussion going on about these types of sponges. See, there's one in the background that doesn't have sort of leaves or knobs on the stalk going up. And then these kind of have these little knobs, and that's the problem. So we're actually not sure.
The great thing about this that I love is the way they're all oriented directly into the current, or is it away from the current? The openings are all pointed in one direction.
Yeah, so they are presumably growing and orienting in the same direction, and I guess the backside, the convex side, would be where the prevailing current is coming from.
Yeah, I think we're done moving.
But I don't know, maybe it isn't.
Yeah, ship stops. You might have a minute or so, but sonar looks clear, so I think turning fine.
This one looks like a Muppet.
Thanks, Sam.
There are some corals interspersed, but they're just overshadowed by this amazing little patch of sponges.
Hey, Chris, is there anything you want to look at in particular?
Right here, I don't think so.
Okay.
Continue that pirouette across the upper side. It looks really nice.
I think as a terrain, this is amazing.
Well,
I mean,
yeah,
I'm
I'm thoroughly enjoying the terrain. I
Yeah.
agree. Just right now.
wow, it goes on and on too.
Co-pilot, can we tilt HD2 up a little bit if that's
That's
acceptable?
why sometimes I think these sponges, when you
Yeah,
come
but
up
can I
with
just
something
release
like
this?
this, you think you're on a different planet. Just so strange.
Yeah, no, Chris, every time we do these dives, all I can think about is, this is the type of experience someone would have if they found, like, life on another planet.
Oh, you want to see it?
Everything's so alien to, at least for me, even though I've been doing it for 5 years, it's still like I'm on another planet when we're down here.
Yeah, and it's definitely the same for me as well, pilot, because even though I work on these sponges, a scene like this is just extraordinary.
And I'll toss my hat in the ring too, as a person who studies echinoderms, which are even more bizarre. These things just stun me. So these things with the two openings, is that Bolosoma or is that Caulophacus?
These are the Bolosomini new genus.
Oh, the new genus.
Yeah, the
Okay.
ET sponge, which of course fits with us discussing alien landscapes.
Of course. But those are essentially filters. I mean, they funnel water current through the head, so to speak.
As you rotate a little bit more
Yeah.
right, I'll light the distance. I think it just continues
So all of
on.
these are arranged in the
Nothingness.
same direction.
It's too bad we don't have any fluorescent dye or something, just to see how the water currents.
Wow.
Well,
that just
typically
goes
the way
on.
a sponge works
I
is
think
that
100 's still a
the
good
water
direction.
goes into the sides of the
If,
sponge.
pilots
you
are
think
happy,
of a
I can
base
put another move
sponge, water would go into
Yeah,
the
I agree with
sides, and then it would go out a large
might be
opening,
approaching
kind of
the top
collects,
here.
and then goes out a large opening through the top.
Yeah,
So you
perhaps.
can think
Not sure
about
which
these
more goes
sponges
up,
the
but we'll
same
see.
way, that the sides or the back
I still
side
got
of this
D2
one we're
sonar
looking at
picking
right
upslope
now,
30
and
meters
the water
out.
is collecting
so.
in it
Okay.
and then going out these two
put 30
larger
meters in,
oscula
100.
that we're
Yeah,
seeing in
sounds
front
good.
of us. So we would be looking up current right now if the interpretation is right. Now, a really interesting phenomenon, and I don't know how it applies
Bridge,
for this
go ahead.
type of sponge, but for base sponges,
sir.
kind of believe, or there's a theory that's been floated for years that they're utilizing
Range,
a
30
Venturi
meters,
effect, so
bearing
that the water
100,
crossing over the
speed
opening
0.2.
at the top of the vase
Shelley,
is
straight
actually
ahead.
pulling water at a faster rate through the walls of the sponge.
Hey,
It's
come
like
on in,
blowing
Will.
through a Coke bottle or something. So
Oh, this is
looks like crinoids all over the
oh, this
place.
little jellyfish too.
Oh, is it? Oh, there he is. I see.
Oh. Ooh.
So is that defensive where he drops?
There's another jellyfish in Monterey Bay called Colobedema, which does something similar. When it feels threatened, the tentacles light up and the bell goes dark. And I think I've read that it's interpreted as a kind of a decoy.
So if,
Yeah,
we,
so
if
they're-
we killed our lights, we'd see glowing stuff?
I think so. That's what the rationale
Right.
was. At least
If we had sensitive enough
ROV
cameras.
nav, move initiated.
You know what this looks like to me from some of the movies? It looks like when you're seeing those war movies where they're talking about dropping countermeasures.
Oh yeah, cool.
All right, come on out.
Chaff, that's right, Matt. Another word for it.
Flares.
flares,
Dropping flares.
whatever it is that distracts whoever's about to eat you so you can get away.
Let's see, this isn't
Is
so
that
bad.
a stock crinoid at the
No.
bottom left?
Oh,
Is that a
here we
stock
go.
crinoid right there?
Sometimes they sneak in
Yeah.
on these filter feeding
All
grounds.
right, come on in.
Very granular substructure.
Oh,
here.
it is.
Yeah, and it looks like it's that one we've been seeing, Bavicrinus, I think. It's got the sort of the same pinnual structure, and the stalk is covered with hydroids. So stocked crinoids are the
Right,
same
come on in.
group of echinoderms as the feather stars that we've been seeing, but they're kind of an older or an earlier form, and rather than sort of living free form, they are connected to the, or rather they're,
This is the bridge. Go
in
ahead.
these cases,
Hey, we had a
the
terrain
discs
range on
are,
us. I'd like to
rather the
adjust,
cups are
our
connected to the
bailout
bottom
direction.
with a stalk.
Roger,
There are
go
some
ahead.
stocked crinoids which
Just
actually have
due
a
west
free stock,
270.
and they can
New
move to a
bailout.
limited degree.
270.
What we're
Copy
seeing
that, thank
here
you.
is where
Thank
all
you.
of the arms converge on the center where the mouth is located. And I believe one of those offset openings might be where the anus is located. But if we move slowly down the stalk, you can see that it's got all of these forms growing on it. Not all crinoids have that. I'm not sure why these have them, which makes them kind of unusual for other, relative to other crinoids. But these are probably hydroids or some other kind of animal, and it's not entirely clear what they're doing there, other than the obvious living and feeding and so forth.
All right, come on out. Turn lasers back on.
Nolan Barrett had mentioned this. Looks like
We can bring those uppers back out.
Maybe Charlie.
Nasako Matsumoto commented that this is ET's home ground or home planet, if you will.
What is that sort of purple silverish bit up in the middle of? Oh, is it just a reflection off the rock? I'm
Yeah, I
sorry,
think
it's
so.
a reflection. Yeah. Wow, I love places like this.
Yeah, so do I. This is why I like the community, to see the whole community and how it evolves over the topography, because you get surprised as you transect.
So,
Just excuse me for a moment.
No,
Pilot,
it's
could you zoom in on that leggy coral that's to the bottom center? I think that's a new record for today
Copy that.
of this one. That's it, right by the lasers.
Come on in, Roland.
Yeah, so this is a primnoid coral, but it's a Narella too, but I think this could be a Narella macrocalyx, which is, I think, the first record, and we got some ophiuroids on it as well. You can see the polyps curling down the stalk, which is typical of that genus Narella, although there's some other genera that can do that as well. But coupled with the very sparse branching coming off near the base of it, I think it's a good guess to go Narella macrocalyx. Thanks very much.
there's
Pilot.
also a little white coral there on the bottom, I think.
What was that, Chris?
Oh, I'm sorry. Near the bottom of the branch of that coral colony, there was an actual cup coral cemented onto the rock. So there was that low branch, and it was just behind it. Yeah, there it is. Kind of
Camouflaged.
hidden behind the branch.
Maybe dead.
Off my
Or did
port
you
side.
see? You saw the
Yeah,
actual
come on
tissue
I
on it?
thought I saw the tissue, but it could have been the primnoid. Oh, I'm sorry, was it primnoid?
Primnoid, yeah.
Primnoid, yeah.
Oh, there's another one of those sponges.
Wow, what's the
Now look at the density beyond here.
ROV
Look
nav,
at this.
move
big
complete.
one on the rock, though? Look at that.
Yeah, that's again just different morphologies of the same weird Fareid Skeletor
Yeah,
sponge.
Copilot, if you want to rotate,
Almost like a Game of
sonar
Thrones
looks okay ahead, so I think you can keep pilot in view until he centers up.
Look at this bizarre
Then we
morphology.
might have another
down
terrain
below us
change
there.
on
Look
us.
at
This
this
looks
yellow.
like a big horn.
So we might have to look around and find a new direction.
Yeah, well, it's just all kind of weird and twisted.
Copilot, permission to zoom your sonar out to 80.
Yeah, go ahead for it, John.
It's almost too much to look at up here.
No, there's never too much to look at, Josh.
We could spend the whole dive in the next, 20 feet.
Yeah.
Probably that direction, huh?
Yeah, you're looking.
So
Yeah,
one
if
of
you
the
want to
things
rotate
we're
more
trying
south
to figure
now.
out here is what
Maybe,
is so special about this spot. Why do we have this incredible
So definitely feel like we're on the
density
top,
of
right?
animals
If we go
here
any
at
further,
this
we'd go
one
over it.
spot
I want to say yes.
comparison to other spots?
So if you want to rotate maybe where
We're
D2 is
just
looking.
collecting data. We're hoping to
That
be able
seems
to
to
answer
be
that question at some
promising
point
for
in the
next
future.
moves.
Yeah, I
I'm going
agree.
to push you back to 50 meters.
One possibility, of course, these are all filter feeding, so current acceleration, sediment
And then we
clear
are about
substrate
30 meters above
depth. They're all among
the
the
last
candidate
contour and high back
factors.
tail.
Cup. Oh yeah, look at
So
that.
we have a request from Tim to get a zoom on some of the ophiuroids that are living on the Narella here.
So we didn't get those already. We got a lot of.
He thinks
Narella?
they're different from the ones that were seen before.
Okay.
I'll keep
we
him in
can do
view.
Yeah,
He comes
as he
over to
tries-
starboard.
Perfect.
I'll
Coral,
turn
a
with him.
fan.
We'll line up.
It's got some opies
I think
on it.
uphill's your
Oh, look,
later.
it's got one of the
You know,
crinoids.
130.
Sounds like a fantastic plan.
Like 130 or
I
something.
remember you
But
telling
we'll
me
see.
the Zygo Metro Day? Or
So
the last one? It's got the little attenuated tips.
well, we used to call this Cerametra crina, something like that. And then Chuck changed his mind about it. Chuck Messing,
Okay.
and I've got to look it up because it's new, right before the cruise. And part of the problem with Cerametra is that it hasn't been documented in the Pacific. Yes, Sathyrometra. It's still in the family Xenometridae.
Phosphate.
Yeah, okay.
All right.
So yeah, as you come up this way, I'll turn with you.
Cup.
Until we find a heading that we want to go. Probably 130, somewhere in there, but figure it out. Forest down here.
Wow.
So I would, I'm going to just throw this out here. Yes, we have plenty of time to go on this dive, but this yellow Poliopogon sponge is just pretty unusual, and I would put it to the group whether or not there's as much interest in collecting one of these as I have.
So that
Again,
one just on
I'm
the top
fine
got
with
two of
moving
them
on.
on the same
But we're seeing
stock.
it. These are weird sponges that are attaching almost exclusively to these dead sponge stocks, which is a really curious behavior.
That one on the
And
top
I mean, not
right, and
behavior,
that got
sponges
two of them on the
don't
same
really
stock.
behave, but, and we've got a different sponge that's to the left of the dead sponge coming down here. I think this is new, but anyway, they're all over the place. I can wait a bit.
Combining roll.
Well, I think it's
He's
considering
referring
that
to the
it's
bottom one that just went off
a
screen.
member of
Yeah.
the community, I'd say that's a fairly good candidate. So I certainly
Yeah.
support it.
Okay, thank you. And after we get this one, this wasn't the sponge I was thinking of. It is, where did it go?
It was
There it
the,
is, to the lower right corner, going off right now.
Kind of the catcher's mitt. looking
Yeah,
thing.
that one right below the lasers now.
Yeah, go ahead in.
Ah, yes, this is what I was hoping it would be. So this is a demo sponge, believe it or not.
Really?
Yes. This is Stellodorix, and
Talk about imposter suit.
We're done moving
Yeah,
now.
Yeah, ship started about 6 minutes
these
ago.
we have
So
collected
you should be started
a
out
few
nicely.
of these
Great, thanks.
over the years. Hurl collected one a number of years ago, and we collected at least one from the Okeanos. And it's this weird branch sponge, and I don't know much about it, and I wish I did. I don't really do much with demo sponges. But yeah, that's a sponge that's assumed a good filtering morphology, I guess.
Sure. Well, as everybody knows, I always go for starfish, but I'm happy to forego a potential slot to collect the sponge because Chris wants it.
Well, it's just looks like there is some support. We don't have to collect this one. I'd like to have one that has maybe we could. Well, that's one of them. All right, that's one of these weird ones here to the right. I wonder if we could get a little better look at it. So
Hey, come in, Roland.
We kill lasers or keep them on?
You can kill.
Going off.
yeah, so this is the guy. It's
It looks like an isopod to me, but if we looked at it from the side, it might be more up-down.
So I think this would.
It's definitely an amphipod.
It is an amphipod. Oh well.
It has on the sort of after the thorax, the first part of the pleon is it's got some spines dorsally. Can't really tell what's going on with the rest of the back end of the body, but the rest of the abdomen is then folded under the forward part. But without getting a better view from the side, you can't really tell which family it belongs to.
Thanks, Les.
Yeah, thank you. Pilot, did you see that one that kind of fell over there?
Mm-hmm.
I think we have a volunteer.
This
That, yeah. I don't suppose he's small enough to fit the whole thing in.
Let's get the lasers on.
That'll fit in an outer.
The bumper. Okay. Yeah.
I think he'll fit. I might have to snip off the dead sponge.
That would be okay.
Hydraulics are up. Ship's stable.
Yeah, this is a good spot to sample.
So he came off pretty easily, which again I don't understand how some of these
Port
aren't
inner is
causing
taken.
more of
Other
these
than
to pop
that,
off.
Port
Unless-
outer is good though.
yeah, I believe so.
Well, it
Sean,
might.
you want to
It
confirm?
might be. That's
No,
why you have
yes,
somebody
the port
laying.
outer is
Yeah,
open.
I mean, or not just laying, but why we have so many bases lying around.
Right.
Looks like there's a crinoid on that base, or on the head of the,
No, in the base of the dead sponge.
yeah, not the actual base, but the dead
The head of the dead sponge.
Yeah, we might be able to get a few things. Oftentimes these sponges have some associates in there. Not just the dead sponge, but the live ones.
Right. See if I can break off one of these
Just to make it.
pieces here.
Yeah, because this might be too big to fit in.
Oh, it's a pretty hard sponge.
I'll try to fit the whole thing in.
Really? What force grip were you using?
6.
Wow. Chris, did you hear that? Chris, did you hear what the pilot was saying?
I'm sorry, no, I didn't. I was
actually used a force grip of 6 on that to try to snap the bottom half, and he could not get it to snap.
Yeah, I guess we tried to. Oh, there goes the Okeanos. We tried to actually take off bottom part of a specimen we took the other day of what I think is similar to this dead sponge, and it was very difficult to cut. It's very durable and hard. So this will be what we call a softer sponge. The actual yellow live sponge itself that's on the dead one does not have the silicified spicules, if you will. Some sponges actually glue their spicules together and make a very hard skeleton like the dead ones. But the yellow one that's on it doesn't do that, so it'll actually feel like a kitchen sponge when you bring it up.
Which did you open? Port. Okay.
I found that really interesting because I thought those brown parts were just super brittle and not very strong.
I'm starting to hit the ground, so I stopped. It's not fully out.
Yeah, maybe it's just the age thing, Roland. It's just that those some of them are very durable and some of them are so old that they've become a little more brittle.
Nice.
Fantastic. That's a remarkable specimen. Thank you very much.
Retracting.
You're welcome.
Easy.
And thanks to the onshore folks that supported that collection. You just don't get very many yellow sponges. We're seeing some today, the yellow bullosomony, the stock sponge, and then this yellow ferronematid, but it's quite uncommon. I think there's some yellow sponges in the Atlantic. Hertwiggia is one example of one, but in the Pacific we don't see that very often.
And we're good on, we can move on here?
Yeah, thank you very much.
So I got uphill, 90 degrees to my starboard pilot.
Okay.
So we'll do a big turn here with me.
Have we seen that pink one at the bottom?
Oh, there's a pink coral in the lower left. I think that's just a corallid, but let's take a look just in case.
Step in.
Thank you. Oh, maybe I don't know. Chris, is that Paragorgia or is that Pamicorallium?
Yeah, Chris.
Thanks. Both? It's pink.
We're seeing the backside, so I think this is a coralliid. Well, maybe we can get a little bit tighter zoom if that's possible.
Yeah, I'm coming in on it.
Max and track and focus.
I think it's a Paragorgia, frankly, but no, maybe not.
It has that rough texture that
Yeah,
hemicorallium
that's
seems to have.
hemicorallium. It's all closed up entirely. Yeah.
Maybe it's the water current that does it, sort of like getting a lot of air in your eye or something.
The way the polyps are closed up is kind of odd to me.
Look at the interesting. Was there a jellyfish on the other side of that? We just scroll up. Yeah, there's like
Where
right
is it?
there. Right
Oh,
there. There's like
yeah.
a. I don't know if that's just some debris or if that's a thing. Huh.
Hydraulics are secure.
I have no idea.
Thank you, Lima.
We're
I'm going to guess Paragorgia on this one because by and large the branches are all thick. And coralliids often have very thin branches, especially out at the ends. And these still seem to be kind of thick. But otherwise, I'm not sure what other characters you might be using, Chris.
Well, I was looking at all the tiny little bumps on the branches, but you know, I'm actually leaning your way, Les, too, because it does not look like hemicorallium polyps closed up, and there can be multiples at the end of the branches too, which is not characteristic of hemicorallium either. And it certainly doesn't look like a pluricorallium that's known. So I think you're right. I think it's a Paragorgia.
Oh, there's another one of those.
Asterophera in the background. If you scroll up to the left. It's just behind the, yeah. Oh, there it is.
Oh, yeah.
Wow. Maybe it's a current thing. Maybe they stay flush in order to stay low because the current would blow them away. Interesting.
Ready for full screen of
We're
this guy?
good, pilot.
Yep.
For that. I think we've gone as far as we can on identifying this thing. So I am agreeing with Les now. It's
So
just
Paragorgia?
yeah, funny. That's how I put it in the annotations.
Okay.
There's a little bit of a head scratcher, but
Yeah, so it's pretty close to me here, Josh. I'd like to turn to starboard, about 90.
Heading your way.
Leave it, Charlie.
so Chris, we're not seeing any urchins today again, which is just
Well, again, I think there's I think there's probably a sediment thing. I mean, the food that they eat isn't here, and there aren't a lot of them that feed on sponges. If anything,
Jelly bottom.
I
center.
expect to see Henricia
And
today.
it's right inside your
Okay.
leaves.
And frankly, I don't want to look at one of those. So.
Let's do a snap here. That's a branch.
I'm not sure who will wind up naming this seamount. Mashkor can figure that out in his mapping team and the OER mapping group, but maybe something that connotes sponges might be appropriate given that spectacular scenery we got.
I think we should call it Asterophera Ridge.
Okay. Well, we had two of those, right?
Yes. No, I obviously I'm kidding.
Yeah, I know you are. But on the other hand, that's pretty cool. It's a cool.
I'm very fond of them, but and the grand sense of things as a habitat, especially like I love the way they're all
It's like
turned,
145
like
for
the,
me.
the convex,
I'm lined
sort of
up,
the concavity
I'm
of
happy.
the bodies is just sort of directed right into the water current. So something like a sponge ridge or They're mostly glass sponges too, aren't they?
Yes, there was that one demo
Yeah.
sponge that we zoomed in a moment ago, but almost all of them are sponges.
But the glass sponges.
Yeah,
Yeah.
right.
Glass Sponge Valley.
Or Hexact Seamount. I don't know. It doesn't quite really roll off the tongue, does it?
Sponge Ridge still has that best ring to it, I'd say.
You know, there's another thing that could be done. You could honor somebody like Henry Ricewig, calling it Ricewig Seamount, because in a sense you're indirectly noting the sponges colonies here.
Well, that'd be very nice, actually.
There's a beautiful corallium colony
is that
there.
corallium?
Yeah, it's
Okay.
corallium. I was just noting it's a beautiful
Any
fan. We
chance
just zoomed
the
in on
front
that.
side
So it
pirouette
may
of
not
that
be
guy?
necessary, but
Which
it's
side's the front?
The right-hand side.
just a
Actually, no, that's the back
gorgeous
side.
fan. So those sponges
is
and
still very
corals are
pretty.
all lined
Killed
up with the
lasers.
same
Lasers coming off?
Partial
direction.
zoom.
Yeah, the
Lasers are killed.
sort of that.
Trying to decide which
You said
end I want to
hemicorallium?
go on.
The
What's
right
that? Yeah,
side would be
hemicorallium.
Yeah,
That's another one.
It's got that parabolic fan that's just kind of
okay to zoom.
bent into or curved into the current. Nice.
I don't think this is the Paragorgia, but we
the
could
colony.
take a zoom in and confirm that. It's just really
All right,
beautiful.
Got any corals specialists that can guesstimate ages?
Not with Paragorgia. This is a question from the
Right.
Roland, the
Come on in,
videographer,
Roland.
about
Okay.
kind of knowing the ages of these animals. We're looking at the backside of this coral, and this looks definitely hemicorallium-like. So that's a little bit more normal.
I mean, just lights
What
are Leo.
these deep corallids
Hey,
look
Leo.
like. And
Okay.
this particular colony could be hundreds of years old for all we know.
Certainly, other corals have set a precedent for being extremely long-lived.
And there has been work on this family in some shallow water members of the Coralliidae, and they've been coming up with between 100 to 200 years for much smaller colonies. So we're good whenever video is, Pilot.
All right, Roland, let me know when you're good.
Okay. You have the time to get all the way around to the front, and we can pull out and go. It's a really nice turn you got going.
Oh, that's nice the way it's coming around. Yeah. Like being spun around on a jewelry turntable.
You can clearly see the difference between the two sides of the sponge. And there's been speculation as to why the coralliids and other species
Pulling
of
out slow.
sponges have this type of polyps on one side and not on the other side, and with the feeding efficiency with having this kind of design. But it's fairly common. There are scleractinians such as Enlapsamia that do the same thing.
Okay. Can take off. Thank you, sir.
It's probably not a coincidence
flying.
that crinoid or that feather star is on that side also. It's probably taking advantage of the current in a slightly different way from the coral, the precious coral
Nice
we
shot,
were
Josh.
looking at as well. So
Thanks.
It's a Chrysogorgia, a little bit different one maybe, that's to the left there. Could have a squat lobster that maybe some people would be interested in.
Go ahead in, Roland.
And there he is.